Welcome to Carbohydrates Video 2 for Chem 314. This time around we're going to talk about glycosidic linkages and disaccharides and polysaccharides. So just to remind everybody, there is a carbohydrate worksheet which is up on Canvas, and we may or may not do any problems from that one in class, but that one will explain basically everything I expect you to know about carbohydrates. So your best practice for doing carbohydrates is to take a look at that worksheet. So last video we talked about monosaccharides. So just to refresh your memory, monosaccharides are the simplest of all carbohydrates. They have 3 to 7 carbons. They have one carbonyl group, and all of the other carbons have one and only one hydroxyl. In today's lecture, we're going to see di, tri, and so forth saccharides, or polysaccharides. And so, what is a polysaccharide? That is a molecule formed from many monosaccharides. I have a very hard time spelling the word monosaccharide for some reason. Connected to each other by what we call glycosidic linkages. So in other words, polysaccharides are polymers of monosaccharides. Well, what's a glycosidic linkage? A glycosidic linkage is what you get when you do this. You take the anomeric OH from one carbohydrate molecule, one monosaccharide, and any OH on a second monosaccharide, and you condense those together, you lose water, and what you get is something that looks like this. Now we have a glycosidic linkage. So you'll notice it looks kind of like an ether. We've got carbon, oxygen, carbon. But the glycosidic linkage has a specific property. One of those carbons, at least, is an anomeric carbon. The other one may or may not be an anomeric carbon. And so glycosidic linkages link the anomeric carbon of one monosaccharide to any carbon of the next monosaccharide. And formation of glycosidic linkages is pretty much irreversible, at least under biological conditions. So it's not like formation of the ring, right? Remember what we said last time is formation of these rings is reversible. Glycosidic linkages are pretty much irreversible, and to break them apart, you need enzymes or acids or some sort of harsh treatment like that. Now, when you make a disaccharide or an oligosaccharide or a polysaccharide, you have to keep track of what type of glycosidic linkage that you have. And we have a specific strategy for naming glycosidic linkages. And I do expect you guys to know how to do that. So the first thing that you have to do is to pay attention to the numbers of your carbon atoms. So on this disaccharide, we have lactose and glucose. Together that makes lactose. And you want to pay attention to the number of the carbons. So remember, the anomeric carbon is always going to be either carbon 1 or carbon 2. In this case, it's carbon 1 on both monosaccharides. All right, and we're not showing the hydrogens just so the figure is less crowded. So what do we see? Well, we see that we have a glycosidic linkage from the anomeric carbon on galactose, which is carbon 1. The OH group is pointing up, so it's a beta anomer, and it connects to carbon 4 on glucose. So this glycosidic linkage here would be called a beta 1 to 4 glycosidic linkage. So it's always going to be something of the pattern alpha or beta, a number, an arrow, and another number. So the alpha or the beta is the anomer of the first monosaccharide or the first sugar. The number is the number of the anomeric carbon. Then you have an arrow, and then the other number is the number of the other carbon that you're connecting to. And if this one is an anomeric carbon, then you also have to put an alpha or a beta there as well. So overall, lactose could be considered the lactose beta 1 to 4 glucose, or lactose connected to glucose by a beta 1 to 4 glycosidic linkage. Let's try another one. What about maltose? Well, first of all, let's find our anomeric carbons. So there's one right there, and there's one right there. Okay, by the way, that squiggly line, that means either alpha or beta. You don't know, you don't care, so we're just going to draw a squiggle. Anyway, number your carbons. Remember, anomeric carbon is always either going to be 1 or 2. So in this case, it's 1 because there's no other carbons outside the ring. 2, 3, 4, 5, 6. On the other one, 1, 2, 3, 4. It points down, so it's alpha. This is an alpha 1 to 4 glycosidic linkage. And so maltose is 2 glucoses connected by an alpha 1 to 4 linkage. There's plenty of practice problems related to this one in the worksheet, and so I suggest you do the worksheet. There's another term that you guys saw in lab, and that's the idea of reducing versus non-reducing sugars or polysaccharides. So we said before that the carbonyl group is reactive, and one of the reactions that it can do is it can reduce copper ions. So a carbonyl with copper can form copper 1 and a carboxylic acid. Yes, you need water in here to provide the other oxygen atom. You're not responsible for this reaction. So in other words, any sugar with a carbonyl can reduce other substances. And that's used as a diagnostic test to see what type of sugar you have. So any sugar with a carbonyl that can reduce other substances, this is what's called a reducing sugar. Any sugar without a carbonyl is said to be non-reducing. Now, if you're paying attention, you're probably thinking, well, wait a minute, didn't you just say a minute ago all sugars by definition have carbonyls? And that is true if they're monosaccharides. But if you have a disaccharide and you form a glycosidic linkage, it's possible that both of those carbonyls will go away. So all monosaccharides are reducing. But if you have a glycosidic linkage, then you may or may not have a reducing molecule. It all is going to depend on where that glycosidic linkage is. So for di, tri, polysaccharides, and so forth, it's reducing if there is an anomeric carbon somewhere that has an OH on it. That's how you tell if it's reducing or non-reducing. So for example, sucrose, this sugar at the right, common table sugar. This is our first anomeric carbon. That's our other one. So notice that the fructose on the right is not drawn in the standard form because the anomeric carbon is on the left. You'll see that neither one of those has an OH because they're in a glycosidic linkage together. Sucrose is non-reducing. And non-reducing sugars are much less reactive chemically than reducing sugars. And that's why sucrose is so useful as a sweetener, is because it doesn't break down and react with other stuff the way that plain old glucose or something like that would, because it's non-reducing. So what about lactose? That's the second sugar in this list. Well, there's one of our anomeric carbons. There's the other one. That one has an OH. Lactose is reducing. Maltose at the bottom, also reducing. I'll leave that as an exercise for you to figure out. Then if you're dealing with polysaccharides, something like this, remember, glycosidic linkages always involve one anomeric carbon. And so in most polysaccharides, you're going to have one end of the molecule that has an OH on an anomeric carbon. So that's called the reducing end, right? Because of that OH. The other end is what we would call a non-reducing end. Now, if you remember the carbohydrates lab, we said that polysaccharides are generally considered non-reducing. Okay, polysaccharides meaning, greater than 3 to 4 carbohydrates long. And we say that because if there's only one anomeric carbon with an OH, but you've got hundreds of anomeric carbons, then honestly you don't have a very reactive molecule. But most of them do have a reducing end, even though we consider them non-reducing, so don't let that confuse you. Okay, so the structure of polysaccharides has to do a lot with the linkages between our different monosaccharides. So we can classify polysaccharides based on function, but we can also classify them by structure. So there's storage polysaccharides, which are used to store energy. So starch and glycogen, for example. And then there's structural polysaccharides, which are used for structure, self-explanatory. Cellulose, chitin, those are examples of that. This is mostly used for things like cell walls, and also in animals, what we call the extracellular matrix, which is that sort of gel of carbohydrates and proteins that's in between your cells to sort of fill in the space because we don't have cell walls. All right, so let's talk about starch first. So starch, it's a mixture of two molecules, amylose and a myelopectin, but they're basically the same thing. It's a polymer of glucose connected by alpha 1 to 4 linkages. So the fact that we have alpha 1 to 4 linkages means that starch has a fairly open and flexible conformation, which allows it to soak up lots and lots of water, which is one of the useful properties of starch in cooking. And so why does it have this open and flexible conformation? Well, remember what we said in video 1 is that your rings are not flat rings, they're actually chairs. And we've got axial and equatorial groups. And in glucose, if you have the alpha anomer, the OH on the anomeric carbon points down, so it's axial. And so that means that each glycosidic linkage causes a bend in the chain, right? And so as a result, if you extend this out over hundreds and hundreds of glucoses, you end up with this sort of open, wide helical conformation, something like this. It's not like an alpha helix in a protein, okay? Do not think it's like an alpha helix, because you'll notice over here that the sugars are so far apart they can't hydrogen bond with each other. So it's this big open structure that soaks up lots of water. You'll notice that in this picture over here at the right, there's iodine, I2 molecules bound in the center of the helix. And if you've ever done that little lab in high school bio where you stain potato sections with a potassium iodide solution, that's what's going on, is the iodine binds to the inside of this helix because it's exactly the same size as an iodine molecule, and that gives you a nice visible color change that lets you know you got starch. The other common polysaccharide that you should know about, this one is a structural polysaccharide, is cellulose. So cellulose is chemically an isomer of starch, but it has radically different properties. And that's because it has beta 1 to 4 linkages instead of alpha like starch. And why that leads to such different properties is because if you have the beta anomer of glucose, the OH is now equatorial. So each successive link in your cellulose chain is basically straight ahead. And so you end up with a straight ribbon-like molecule, and the different chains of cellulose can then pack side by side and hydrogen bond with each other, forming a structure that is actually a lot like a beta sheet from a protein. You've got the different cellulose chains hydrogen bonding with each other side by side. And then these can also stack on top of each other, giving you this nice three-dimensional rigid matrix type structure. So for that reason, cellulose is very rigid. And even though it's very polar, it's insoluble in water because of all these hydrogen bonds. There's a similar, less well-known structural polysaccharide called chitin, and chitin is almost the same thing as cellulose. It's also got beta 1 to 4 linkages, but instead of glucose, it's in acetylglucosamine, which means that instead of this OH group over here, what we have is NHCOCH3. So that's chitin down there. So because of that big bulky side group there, we end up with a less rigid molecule compared to cellulose. And that's because that big bulky N-acetyl group hanging off of the side kind of impairs packing side by side, and therefore you get much less hydrogen bonding. So chitin is found in the exoskeletons of insects. It's also found in the cell walls of fungi. And in both cases, it's a bit less rigid than cellulose, which is what's found in the cell walls of plants. And structural polysaccharides are not just for plants and fungi. We have them too. The animal equivalent of these are what we call the glycosaminoglycans, or GAGs. So what's a glycosaminoglycan? So these are polysaccharides that have disaccharide repeats. So in other words, the same two monosaccharides repeated hundreds or thousands of times. They always have a strongly negative charge, and they always soak up water from their surroundings. And the reason they soak up water is because of that negative charge. So these things tend to form very extended chains like that because of the charge repulsion among all the different negatively charged groups. So we end up with an extended conformation in water. Now, depending on what particular sugars you have and what type of glycosidic linkage you have, you've got all different types of GAGs, and it's just totally pointless to have you know them all. So don't even worry about it. Just know what they are and just know what they do. So for example, hyaluronan, which is found in cartilage, you've got roughly 50,000 sugar repeats per molecule. You've got a gluconic acid, a beta 1 to 3 linkage, an N-acetylglucosamine, a beta 1 to 4 linkage, and then you repeat. So notice you got one negative charge per repeat, so that's not super negatively charged. And then there's others. There's chondroitin, there's keratin, I should say. Keratin is a protein, don't get those confused. Heparin is super negatively charged. That's what it looks like in 3 dimensions. So it's negatively charged because of all these sulfate groups everywhere. So if you have blood drawn, you'll notice that in the tube, before they fill it with blood, they put in a little yellow slime at the bottom. That yellow slime is heparin. And what heparin does is, because of all these negative charges, it binds up calcium ions out of your blood. And since blood clotting requires calcium, it prevents blood from clotting. That's why they mix it in with your blood sample. So all you got to know about GAGs is they are disaccharide repeats with negatively charged groups and extended chains. They bind to proteins and they soak up water. And that's what we have filling in the gaps between our cells. So that mostly exhausts the subject of carbohydrates. What we care more about than the chemistry of carbohydrates in this class is the uses of carbohydrates. So our next unit, the metabolism unit, is where we're going to look at oxidation of carbohydrates to get energy. And that's the reason that we introduce carbohydrates at this point. So you have that to look forward to in the next unit of this course. In the meantime, once again, check out that carbohydrate worksheet, and I'll see you in class.